Assalamualaikum and very good morning to Dr. Mariam. We will be presenting a poster of research paper entitled Hepatofauna in Southern Part of Pulau Tioman, Pahang, Peninsula Malaysia. This presentation will be presented by me, Nadira, Sahira, Farhana and Naklia. Pulau Tioman is the largest island that reached the highest peak of 1.035 meter, which is Gunung Kijang. It also has various zones of vegetation such as mangrove, coastal vegetations, lowland dipterocarp forest, hill dipterocarp forest, and rich forest. From the previous study that have been recorded that 23 species of amphibian and 74 species of reptiles. The aim of this study is for documented the diversity of hepatofauna into small area in southern part Pulau Tioman.
Hi, my name is Sahira. Today I'm going to present the study sites and the material and methods of this study. The Hepatofauna survey was conducted for 5 sampling days at 2 areas in the south region of Pulau Tioman, which are Kampung Asar and Kampung Mukut. The study area at Kampung Asar consists of areas at the base camp, trekking trail heading to Kampung Mukut and Sungai Asar. The sampling area at Kampung Mukut consists of human settlement area and the nearby river, Sungai Raya. During the inventory, 2 methods were carried out, which are visual encounter surveys to systematically search for active terrestrial and arboreal species, while cage trapping employed to focus on aquatic species. The species search of visual encounter survey consists of diurnal and nocturnal surveys, with the headlight for visual aid during nighttime. Visual encounter survey was done just after the nightfall, which is 7:30 p.m. and lasts for 4 hours, involving 2 to 5 persons. 3 fish traps were set up in order to sample aquatic reptiles in the small streams at the study area. The representative individuals were collected for species identification and photograph. The samples were euthanized and fixed with 10% of formalin and transferred to 70% of ethanol for long-term storage. All of the collected specimens were deposited in the Institute of Biodiversity, Department of Wildlife and National Park Lancang, Pahang.
A total of 30 species was discovered in this study, which comprised of 8 amphibian species and 22 reptile species from 2 nearby locations at the southern part of Pulau Tioman, which was Kampung Asar and Kampung Mukut. The hepatofaunal diversity in Kampung Asar was higher than Kampung Mukut, which was 25 species and 11 species respectively. These bring the total species known for each location to 28 and 27 species. For amphibian, there were 4 families, 2 species of toads and 6 species of frog. 8 families of reptiles, which was 7 species of lizard and geckos, 3 species of skink, 1 species of soft-shell turtle, and 4 species of snakes were recorded.
Discussion. The number of species found in Kampung Asar was higher than Kampung Mukut, which is Kampung Asar has 25 species while Kampung Mukut has 11 species. However, disturbed environment in Kampung Mukut provided many breeding sites for commensal species and foraging ground for insect for scansorial animals. Next, disturbed habitats in Kampung Mukut support common endurance. Disposal of food waste and garbage in human villages seem to attract scavenged reptiles such as water monitor lizard. Dense undergrowth such as shrub and low vegetation also provided sheltered and foraging ground for diurnal reptiles. Most of the species recorded in this study can be found elsewhere in Peninsular Malaysia except several species which were endemic.
In conclusion, there were 40 species of herpetofauna that have been recorded that consist of 10 species of amphibian and 30 species of reptile from these two areas. Kampung Asa support more species of herpetofauna compared than Kampung Mukut. No additional species or new record that have been recorded in this study. Lastly, these two locations in southern part of Pulau Tioman has rich herpetofauna diversity and should be guidance against any further disturbance.